Topic 4 is the evaluation of averages between samples, under the assumption that the values from the samples are normally distributed. There are 5 subtopics. 1. Describe the differences between parametric tests and their application scenarios. 2. Perform statistical tests for equality of variances. 3. Perform one-tailed and two-tailed tests on 1-2 means for samples using z-test and t-test as appropriate. 4. Apply one-way ANOVA test for single-factor experiments. And 5. Apply two-way ANOVA test for two-factor experiments. Many statistical tests are to compare whether one sample differs from another. Is there a difference between the average height of males versus the average height of females? Is one class better than another class? Then, what we are really asking is whether the average marks for class A more than or different to class B. Similarly, is aerobics helpful in reducing waistline? This question can be translated to whether participants of a 10-week aerobics program show reduced waistline as compared to the start of the program. Does adding extra salt improves the taste of my meal preparation? How to do this experiment? Perhaps we can have 2 identical recipes, one with normal salt content, another with 10% more salt. Then we have 2 groups of tasters to evaluate and examine their average scores. In other words, what we are trying to measure and quantify differences of means. Are the means significantly different? Does the increased salt preparation group give higher average score than that of normal salt preparation group? Let's take for example we want to know whether there is a difference between the average height of males versus the average height of females. Let's say we have 2 random samples of males and females with 100 participants each and we measure all their heights. Assuming that our result suggests that the average male height is 175 centimeters with a standard deviation of 5 centimeters while that the average female height is 168 centimeters with a standard deviation of 5 centimeters, can we say that the average height of males is significantly different from the average height of females from the alternate hypothesis point of view? Or from the null hypothesis point of view, the population mean of male heights equals to the population mean of female heights. Taking this more generically, the null hypothesis is that the values for the height of males and the height of females derives from the same population. Then for the alternate hypothesis, the values for the height of males and the height of females derives from the different populations, in a generic sense. Therefore, you can consider that parametric tests are mainly used to compare the statistics from 2 populations, by using 2 samples, originates from the same population. Average or mean is a statistic of interest here. This can be extended to more than 2 samples. However, parametric tests are based on the assumption that the populations are normally distributed, and you can test this assumption using Shapiro-Wilks test, which is covered in topic 2. Before we can test whether the means of 2 populations may be equal by evaluating the means of their resulting samples, we have to know whether the variances of the 2 populations can be assumed to be equal. This also implies that we must know whether the standard deviations, since standard deviation is just the square root of variance, can be assumed to be equal. Variance plays a critical role in determining whether 2 means are statistically significant or not. Why? Assuming that we want to check whether the average weight of a packet of instant noodles is 80 grams is labeled. And I call this type of questions as quality control questions, is the average weight of a bottle of something matches the specification. It has implications in consumer confidence to more serious case, life and death. Think about it. What will happen if the label of tablets says that it contains 200 milligrams of medicine when in fact, it contains only 180 milligrams or 220 milligrams? Well, depending on the medication, it may result in overdose or ineffectiveness. This is the kind of questions in Laboratory 5 exercises. Let's go back to our question. Assuming that we want to check whether the average weight of a packet of instant noodles is 80 grams is labeled. So, we purchase 50 packets of instant noodles and weigh them. This is the experimental step. We found that the average weight is 78.5 grams, so is the manufacturer cheating the customer? Well, that depends on the variance or standard deviation. Why is that? Let me explain it graphically. Assuming that there are two QC teams for this study. Both teams found that the average weight for a packet of instant noodles to be 78.5 grams. However, the standard deviation of Team A's data is 1 gram while the standard deviation of Team B's data is 0.75 grams. Then we basically ask, what the probability is area under the graph where mean weight is equal or more than 80 grams. We will need to look at the area shaded red. Team A will say that 7.2% chance that the mean weight is equal or more than 80 grams. Since there is more than 5% chance that the mean weight can be equal or more than 80 grams, the difference is not statistically significant. This means that based on the Team A's data, we cannot say that the manufacturer may be cheating the customer as getting an average weight of 78.5 grams may be a sampling error. However, Team B will say that there is 2.6% chance that the mean weight is equal or more than 80 grams. Since there is less than 5% chance that the mean weight can be equal or more than 80 grams, the difference is statistically significant. This means that based on the Team B's data, we can say that the manufacturer may be cheating the customer. Hopefully, you can see why variance or standard deviation is important. So how do we test whether the two variances can be assumed equal? We have to go through the 6 steps of hypothesis testing. Step 1. State the hypothesis. Null hypothesis is that the two variances are equal. Alternate hypothesis is that the two variances are not equal. Step 2. Select the alpha. Here, we set the alpha to equal to 5%. Step 3. What test to use? In Excel, you can use F-test2 samples for variances in Data Analysis Toolkit. In R, we can use Bartlett test or Fleiner-Killeen test. We are going to use Bartlett test, which you will be doing in Laboratory 6. Step 4. Performing the test. In this case, we are testing whether the variance of glycosylated hemoglobin of male patients can be assumed to be equal to the variance of glycosylated hemoglobin of female patients using Bartlett test. Bartlett test gives us the p-value of 0.2671. Step 5. Accept or reject null hypothesis. Since there is less than 26.7% chance, which is higher than the alpha of 5%, that the null hypothesis is true. We fail to reject the null hypothesis. Hence, we accept the null hypothesis. Step 6. Interpret the results. Therefore, the variance of glycosylated hemoglobin of male patients can be assumed to be equal to the variance of glycosylated hemoglobin of female patients. And this is how we can test whether two variances can be assumed equal. Next, we come to the fundamental core for hypothesis testing of means. That is, are two or more means or averages statistically significant or not? Here, we look at one or two means first and the statistical tests to use are z-test and t-test. However, we have to sort out another term before that. In more manual way of doing statistics, the pen and paper way, we have to consider whether we are looking at one-tailed test or two-tailed test. In one-tailed test we are looking at whether the average of a more than average of B, or whether the average of a less than average of B. But in two-tailed test, we are examining whether the average of a different, not equals to, average of B. Quite often, we use two-tailed test. In Excel, you may be confused with one-tailed or two-tailed test. In R, it is almost always two-tailed test or whichever case that makes more practical sense. We can have one sample test, which is what I called as quality control or QC questions. Does the mean statistically differ from a specific value? The null hypothesis takes the form of mean of population equals to x, where x is some value. In fact, our instant noodles example previously is a one-sample test. More specifically, a one-sample z-test. Why it is a one-sample test? Because it asked whether the mean weight of a packet of instant noodles equals to a specific weight, and in this case, 80 grams. Why is it z-test? Well, z-test is for large samples, and statistically speaking, when sample size is more than 30, it can be considered as a large sample. If sample size is less than 30, then we should use t-test. In fact, t-test is meant for small sample sizes. So you can see that z-test is not very useful in a practical sense since it requires large sample size. Yes, we seldom use z-test. Actually, it is not entirely wrong to use t-test even when sample size is large, and many people uses t-test even when z-test can be used. In fact, as the sample size increases, t-test behaves more like z-test. You will do one sample z-test and one sample t-test in Laboratory 5. Next, we come to two samples test, which I call as versus question. There are two types of two samples test. Statistically, we call them as independent samples test or dependent samples test. Here, we look at independent samples test first. Versus question basically means A versus B. Is there statistical difference between mean of sample A and mean of sample B? The null hypothesis takes the form of mean of population A is equal to the mean of population B. For example, is the average marks of group A equals to the average marks of group B? It is called independent samples because group A and group B are independent from each other. For example, I want to know whether is there a difference between two teaching methods and allocate students to either group A or group B. There are 10 students in each group, and here are their test results. Since the sample sizes are less than 30, we can only use t-test. First, we have to check whether the variances can be assumed equal. If yes, the variances can be assumed equal. Then we can use t-test assuming equal variance. If no, the variances cannot be assumed equal. Then we must use t-test assuming unequal variance. For simplicity's sake, I will use f-test two samples for variances in Excel. Results show that since p-value is 0.162, we fail to reject null hypothesis. Hence, we accept null hypothesis. Therefore, the variances can be assumed equal, and we can use t-test assuming equal variance. Finally, we can answer our main question: is the average marks of group A equals average marks of group B? Again, since the variances can be assumed equal, we can use t-test assuming equal variance. Results show that since p-value is 0.959, we fail to reject null hypothesis. Hence, we accept null hypothesis. Therefore, the average marks of group A is not statistically significant to the average marks of group B. And that is how we can perform independent samples t-test. The same steps apply for independent samples z-test, but checking equality of variance is not needed as z-test does not have different versions for equal variances and unequal variances like in t-test. The second type of two samples t-test is known as dependent sample t-test. This is also known as paired t-test. There is no dependent samples version for z-test, so we just focus on dependent sample t-test. It is called dependent samples or paired samples because there is a pairing. Very often, it is the same test subject before and after. For example, You want to evaluate the effectiveness of a 10-week exercise program in terms of weight loss. So you are then asking, is there a difference in weight after 10 weeks of physical exercises compared to before? So you take before and after weights of each participants, which implies that the pairing variable is the participant. Another example that we like to do is whether revision improves students' results. How do we do that? Null hypothesis is then the average difference of marks before and after revision equals to 0. You might realize that this null hypothesis sounds like a quality control or one-sample hypothesis. Does the mean statistically differ from a specific value? In fact, it is the same. Dependent samples t-test is reduced to one-sample t-test. Anyway, we have the before revision marks and after revision marks for 10 students. And we can do a paired t-test. Our paired t-test results show that since p-value is 0.034, we reject null hypothesis. Hence, we accept alternate hypothesis. Therefore, there is a significant difference, from average of 64.6 marks before revision to average of 68.7 marks after revision, suggesting that the revision is helpful. And that is how you do paired t-test. With this, we covered the important tests for 1 and 2 samples. What happens if we have more than 2 samples? That is the next subtopic. T-test or z-test can only compare up to 2 samples. What will happen if I want to compare more than 2 samples? For example, let's say I have 3 recipes of curry chicken and I want to evaluate whether there is a preference for a particular recipe. This is where ANOVA comes in. ANOVA can compare more than 2 means. ANOVA is the acronym for Analysis of Variance. Hold on, we are comparing the means, so why are we analyzing the variance? If you recall our earlier slides on equality of variances, variances are critically important. ANOVA can go from 1 way to 2 ways to as many ways as you want. There is no limit. The way, or factor, represents how many ways you can dice the dataset. Let's look at just one-way ANOVA. In all one-way ANOVA, the null and alternate hypotheses are the same. Null hypothesis is the means of all samples are equal. The alternate hypothesis is at least one of the means is not equal. Take note here, if you reject null hypothesis and accept the alternate hypothesis, ANOVA does not tell you which of the means is different to the rest. It does not even tell you whether only one mean is different from the rest or 2 or more means are different from others. Lastly, ANOVA assumes that all variances are equal. So let's use our recipe question here. I have 3 recipes of curry chicken and I want to evaluate whether there is a preference for a particular recipe. I have 30 tasters in total and each taster evaluate a bowl of curry chicken and score from 1 to 10, with 1 being the worst and 10 being the best. The null hypothesis is the means of all samples are equal. The alternate hypothesis is at least one of the means is not equal. So let's perform the test using Excel and see the results. Our result says that since p-value is 1.6 times 10 to the power of -7, which is incredibly small, we reject null hypothesis. Hence, we accept alternate hypothesis. Therefore, at least one of the recipes is better than the rest. We do not know whether one recipe is better than the other 2 or all recipes are different. However, we can look at the average scores of each recipe and deduce that likely all recipes are different and the best recipe is recipe C. There are other statistical tests to determine whether one recipe is better than the other 2 or all recipes are different, but this is out of the syllabus. You can go and read up on post hoc tests, such as Tukey's HSD, by yourself if that is required. Nevertheless, this is how you perform one-way ANOVA. You can consider one-way ANOVA to be an extension of t-test for more than 2 samples. That is all that is. However, Two-way ANOVA is where the interesting part begins. You can consider two-way ANOVA as a massive extension to one-way ANOVA, so massive that we classify two-way ANOVA, three-way ANOVA, and so on, as n-way ANOVA, as opposed to one-way ANOVA. Let's illustrate with an example using our example in one-way ANOVA, the curry chicken recipe evaluation. In one-way ANOVA, you know that each recipe is evaluated by 10 tasters or evaluators. Here, I tell you that each recipe is evaluated 10 tasters or evaluators comprising of 5 males and 5 females. What other interesting hypotheses you can consider? What other interesting hypotheses you can consider besides whether the average ratings of each recipe is statistically different or not? We can have 2 more hypotheses, making a total of 3 hypotheses. The first hypothesis is the hypothesis we tested in one-way ANOVA. There is no difference in ratings between the recipes. The second hypothesis is that there is no difference in average ratings given by male evaluators versus female evaluators, regardless of recipes. In this second hypothesis, we look at the average of male ratings versus the average of female ratings, regardless of recipes. The third hypothesis is that there is no interaction between the first and second factor. That is, there is no interaction between recipes and gender. This is known as the interaction factor between the 2 main factors. Or in practical terms, do males and females prefer different recipes? Excel is very limited to perform two-way ANOVA and definitely cannot perform three-way ANOVA or more. So we have to use R. However, in order to use R, we have to change the format of the data. There are 3 results since we have 3 hypotheses. Firstly, for gender, with null hypothesis as there is no difference in average ratings given by male evaluators versus female evaluators, regardless of recipes. The p-value is 0.491 and we fail to reject null hypothesis. Hence, we accept null hypothesis. Therefore, there is no difference in average ratings given by male evaluators versus female evaluators, regardless of recipes. Secondly, for recipe, with null hypothesis as there is no difference in average ratings for each recipes. The p-value is 6.45 times 10 to the power of -7. We reject null hypothesis. Hence, we accept alternate hypothesis. Therefore, there is difference in at least one of the average ratings. This is the same conclusion we get in one-way ANOVA, so no problem here. Lastly, for gender with recipe, we are asking whether males and females prefer different recipes. Since p-value is 0.754, we fail to reject null hypothesis. Hence, we accept null hypothesis. Therefore, males and females do not prefer different recipes, which means that there is no interaction between gender and recipes. If this interaction factor is significant, it may tell us something about taste preferences between males and females, and it may have implications to how you want to run your restaurant. Nevertheless, that is how you can analyze two-way ANOVA. And with that, we come to the end of topic 4. Recall that parametric tests in this topic assumes that the data is normally distributed, so what happens if the data is not normally distributed? That is the materials for topic 5.